Welcome back, everyone. It's Dr. Lindner, and we are continuing with neurology. And what I'd like to talk a little bit about now is the neuron. If you think back to Anatomy and Physiology 1, this was discussed briefly when you covered the 4 types of tissues. When you covered the 4 types of tissues, you discussed epithelial tissue, connective tissue, muscle tissue, and nerve tissue. When nerve tissue was discussed, hopefully you focused a little bit of time and energy on the neuron. The neuron is the functional unit of the neural system, and it's the only type of cell that we have that's electrically excitable or that can produce action potentials. Now, I will send out a video later this week. It's also on my YouTube channel that goes over electrical physiology and neural physiology of how action potentials are generated in relationship to sodium and potassium, resting membrane potential, threshold, depolarization, repolarization. You're going to hear terms like that. So it is in one of my videos. Okay, so when we talk about a neuron, we do have some main parts like a cell body, also referred to as a soma. There is a nucleus inside of a neuron, and there are nissl bodies. Nissl bodies are like rough ER and ribosomes that are needed to produce proteins. And the proteins that are being produced by neurons, I would be thinking along the lines of neurotransmitters, right? Neurotransmitters are proteins. Enzymes are proteins. Neurotransmitters like acetylcholine that you learned in AMP1, acetylcholine was really important when we were talking about the neuromuscular junction where the axon hit the muscle and there was the synaptic cleft, and you spoke about the vesicles that were released into the synaptic cleft, acetylcholine being released by the activation of calcium, right? That whole complex. So nissl bodies are kind of important. We have cell processes, predominantly dendrites and axons. When we look at a neuron here, we can see that we have dendrites, which comes from the Greek word dendra, which means branches. So these are the branches of a tree. And then we have a region here called an axon hillock, which is this kind of dilated triangular part of the axon. But the axon is all of this, right? It's everything distal to the axon hillock. It's all of this. And then right at the very end is the axon terminal, and you have those swollen end bulbs, those synaptic end bulbs, which are where those neurotransmitters are kind of stored, okay? So we have dendrites, we have an axon hillock, we have axon. What else do we have here that's important? These nissl bodies, those nissl bodies are important. And notice that the dendrites are not myelinated. What I mean by that is myelin is what we see here. I'm circling it here where it says myelin sheath. It's this insulation or protection that is actually created by a neuroglial cell called a Schwann cell. So the Schwann cell is producing myelin, which is made up of like fat and cholesterol and lipids, and it's an insulator that protects the axon. It also speeds up the action potentials. So when the action potential is generated and it moves down the axon, the fact that it's insulated by myelin, it really speeds up the action potential. It increases the impulse speed. Now, each of these Schwann cells, you'll see in between each Schwann cell is a slight little gap called a node of Ranviere. So this is a node of Ranviere, this is a node of Ranviere, this is a node of Ranviere. The nodes of Ranviere is that small little unmyelinated section of the axon, and nerve impulses actually leap from Schwann cell to Schwann cell. It leaps, which is involved in speeding up the action potential. They call that saltatory conduction, and saltatory in Latin, saltare, means to leap or to jump, saltatory conduction. Okay, let's see what else we have here. So the dendrites, those branches here are conducting the information towards the cell body, okay? And the axons carry information away from the cell body. So I typically tell my students, remember the A in axon with away. The A in axon for away, meaning the axon will conduct impulses away from the cell body, whereas dendrites are bringing the information to the cell body. The dendrites are typically unmyelinated, whereas the axons should be myelinated. Again, what's doing the myelination? What's doing the myelination are these Schwann cells. The Schwann cells are a type of neuroglial cell or neuroglial cell. The term neuroglial or neuroglial means nerve glue. It's holding it all together, these neuroglial cells. And you'll see that here in the center, this is the axon, and it is engulfed by a Schwann cell that keeps wrapping around and wrapping around and insulating and insulating and insulating the axon. Okay, again, you can see here is the axon, and it is completely engulfed and protected by the myelin sheath. Now, if you break down the myelin, what we call that demyelination, when you demyelinate, there's a disease known as MS, which is multiple sclerosis. Multiple sclerosis is when there's an autoimmune condition against the Schwann cells in the peripheral neural system or the oligodendrocytes in the central neural system. Both Schwann cells and oligodendrocytes both produce myelin, but it's like real estate, right? Real estate is location, location, location. Schwann cells myelinate in the PNS, whereas oligodendrocytes myelinate in the CNS. Oligodendrocytes myelinate only in the brain and the spinal cord. Whether there's an autoimmune attack against Schwann cells or oligodendrocytes, it results in demyelination, and it's called multiple sclerosis. Okay? You can see at the very, very end of the axon are these end bulbs, and they're filled with these neurotransmitters. The big important one is acetylcholine, especially when we're talking about the neuromuscular junction. And at the very end, this is what you learned in A&P1, where you would have some sort of skeletal muscle here, and there has to be a way of transferring that neurotransmitter into this muscle to make it contract. And that's where you learned about actin and myosin and troponin and tropomyosin and how calcium came in and stimulated the release of acetylcholine, and there was a whole cascade of events that took place there. Okay, here's just a few histological questions that I've seen pop up in the past on some of the McGraw-Hill Connect quizzes. So when you look at this highlighted structure, right, the red going across here, this is skeletal muscle, and the blue portion is the axon of a motor neuron, okay? The motor end plate would be this. That's the motor end plate. And then the skeletal muscle fiber, well, that's the red fibers running horizontally. This is another histological slide. You'll get used to seeing these at first. You're like, what am I looking at? But what we're looking at here are these synaptic vesicles, these yellow structure. These are synaptic vesicles in a presynaptic terminal. All right. The synaptic cleft, that's this right here. This space here, that gap is the synaptic cleft. In fact, here, the highlighted structure in yellow, that would be the synaptic cleft. All right, now here, this entire structure is a neuron, but the blue portion is the cell body or the soma of the neuron. If we looked here within the cell body, these little speckles, these are referred to as Nissl bodies. These Nissl bodies are going to be making those neurotransmitters and proteins. If we looked at the highlighted section here, the branches off of the soma or cell body, those are going to be dendrites. When we look at the dilated portion, that's somewhat triangular, right off of the cell body just before the axon, well, that's the axon hillock. If it was this portion here, then that would be axon. Okay, now we're looking at the yellow, the external yellow portion. That's that cholesterol and lipid and fat, that's myelin sheath. Now, what's on the inside of that myelin sheath right here? Well, that's the axon. It's a myelinated axon. The myelin is what we saw here. So inside, that's going to be the axon, but it is a myelinated axon. And then this is a Schwann cell, right? This is a Schwann cell. This is a Schwann cell. And the spaces between each Schwann cell, that's what we call the node of Ranviere, where the action potential skips over and only travels along the myelinated portion. In terms of classifying neurons, we have a sensory neuron, we have a motor neuron. You heard me in a previous video mention the sensory and afferent are together. Remember, if this is the brain and this is the spinal cord, anything coming up into the brain is sensory or afferent, and anything that starts in the brain and goes down, that descends, that's motor or efferent. Now, interneurons are the bridge. It's the bridge that connects the sensory to the motor. So in this case here, what we're looking at to the right is a cross-section of the spinal cord. The letter H in the middle is gray matter. It's unmyelinated. The section around the butterfly, that's all myelinated. That's called white matter. And you'll see that right here in starting position number 1, there's a pin or a nail hitting the skin. Ouch, right? There's a pain receptor. Now, this receptor is not feeling any pain. It's picking up the stimulus. That receptor is going to send information into a sensory neuron. A sensory neuron. It's going to send it right into the dorsal horn. This is a dorsal horn. In the front, it's a ventral horn or an anterior horn. So the gray matter has horns. It has a dorsal horn, and then it has a ventral horn. That's the gray matter. In the white matter, we have dorsal white columns. That's white matter. And then in the front, we have ventral white columns. So you have dorsal horns and ventral horns. That's gray matter. And then you have dorsal columns or dorsal white columns and ventral columns or ventral white columns. That's myelinated. So sensory neuron comes in to the dorsal horn, and there's a synapse, a relay, if you would, and it's going to relay that information to an interneuron. This in the center, right here, that relay is called an interneuron. An interneuron. Also referred to as an association neuron. So what we have going from here to here, this bridge here, this is an interneuron or an association neuron. And all it's doing is that it's taking that sensory input and it's going to relay it to the motor neuron. So sensory input comes in, relays it in the gray matter to the interneuron, sends the information out to a motor neuron. The motor neuron goes to the muscle, makes it contract or pull away from the painful stimulus. Okay, now I think you heard me earlier when I talked about the CNS, central neural system, and the PNS, the peripheral neural system. One of the things that I mentioned in the peripheral neural system, besides cranial nerves and spinal nerves, was a ganglia, and I referred to it as a dorsal root ganglia. Well, right here, this sensory neuron comes in the dorsal horn, so it's in the posterior, and this right here, that's the DRG, the dorsal root ganglia, right there. Sensory information comes right in. If you look at it, look at that neuron, it's kind of like this, has a little extension, and then it has this swollen little portion to it. That's a dorsal root ganglia. Okay? So sensory neuron, motor neuron, and in between them, interneuron. Sensory comes into the back to the dorsal root, and motor comes out from the front in the ventral root. So this is a ventral root, and this is also a dorsal root. A dorsal root. Now, structurally, how are they classified, right? This was functionally how they're classified, sensory motor or interneuron, aka association neuron. The structural classification is by the appearance. So when you look at this one here, which is the majority and the most common cell type in the neural system, that's called multipolar. It's when you have several dendrites with one axon. And that's what we've been accustomed to looking at, right? That's your multipolar. Over here in the center, this is bipolar. A bipolar neuron is when you have one main dendrite, which we have here, right? One main dendrite coming off the cell body, and you have one main axon coming off of it as well. That's called bipolar. These are found with your special senses, like in the eye, the retina of the eye, the inner ear, and sense of smell in the nose for olfaction. That's bipolar. Now this one here, the unipolar, this one is always sensory. Now if you look at this, but let's say I put it horizontal. If I put it horizontal, it looks like this. And where did we see this unipolar neuron? I think we saw it right here. Right? I drew it out. That's the DRG, or dorsal root ganglia. And what information is it carrying? Sensory input. Here it was pain. So right here, the unipolar is always a sensory neuron, and the classical example is the dorsal root ganglia. What we're looking at here, dendrites, cell body, axon, That's our multipolar neuron. Okay, and again, you can review, it matches up here with this structure. Other terms are association or interneurons, and some of these are named after the histologist, or sometimes they're named by their appearance. So Purkinje is named after the histologist, but pyramidal, because pyramid, if you look at the cell body here, these look pyramid shaped. Okay, so when you read about some of these fibers, you have a little bit of familiarity as to how these things earned their names. Okay, okay, good place to take a break. When I come back, I'll talk about, now that we've covered the neuron, we'll talk about neuroglial cells, the neuroglial